I then start? Yeah, okay. Good evening, everyone. Today, I'd like to introduce you to the fascinating phenomenon, at least for me, of replication-dependent and independent DNA repair. It sounds weird, right? But why study DNA? Why is it important? Because basically, DNA is everywhere. It consists of two strands or strings of information in the form of four-letter code, A, T, G, and C, which determines the shapes and also functions of all living things currently known to us. And this information, this vast information, is further organized into smaller units or fragments called genes, which are associated with particular tasks. You can think of DNA as being this long information-rich text which consists of individual paragraphs or sentences which convey certain meanings, and these are the genes. And depending on what parts of information, which piece of information or gene a cell uses, it can adopt widely different functions and also shapes. For example, if you use that red bit over there, you have a red cell, blue bit over there, you have a round blue cell, and that bit over there, you become a green cell. But then, as you can imagine, for such a crucial process, this process is tightly regulated. So, in order for a cell to be able to extract accurate information, when it is required to do so, two things need to happen correctly. First one, the cell needs to extract the information correctly, right? So what happens is this information in DNA is transcribed, the jargon is, or copied to an intermediate molecule called a messenger RNA, and in turn this messenger RNA will be translated or decoded, so to speak, into proteins. And these proteins are what enables the cells to do their functions. So basically, what makes our red cells red and our blue cells blue. And secondly, what needs to happen is this. So when a cell divides and produces offspring or children, they need to be able to faithfully copy or replicate their DNA. As you can see here, the upper, top and lower strand of the parent is used as template to generate new strands of DNA, which can then find their ways to the daughters. And this process of DNA replication has a characteristic structure. It's called a replication fork, which consists of two old strands and two new strands, and I will come back to that later. But this process needs to happen correctly if the cell wants to pass its information accurately to its offspring. In order to appreciate the importance of maintaining an accurate DNA code, we can look at what happens if there is an alteration, a change in the code, unwanted change. If, let's say, you have this change in one letter, even one letter, seemingly insignificant, what will happen? It can change the information, the whole information containing the gene, and now the cell produces wrong proteins. And as a result, the cells behave differently. For example, they can now divide uncontrollably and become cancerous. So you don't want to have that. But tonight, I'm going to talk about a specific type of DNA damage which can cause, again, changes in the code of DNA resulting in unwanted consequences. It's called an interstrand crosslink, or ICL. It happens when this cross-linking agent over here joins the DNA together too strongly, or the chemical jargon is covalently bound. So, as you can, I think I showed you before that actually the DNA needs to be separated and it needs to be copied, it needs to be, the information contained therein needs to be accessed, right? If they can't be separated, what happens? You have problems, a lot of problems actually. here, for the purpose of this talk, I will present the interstrand crosslink in a diagram like this. Okay, so as I said, if you have an interstrand crosslink, you have a big problem. Why? Because let's say here, in the case of when you have an interstrand crosslink and you need to extract the information from DNA, you can't because it blocks, right? And then you have no messenger RNA, you have no protein, you have problems. And then again, if you need to copy the DNA, you need to separate it and copy the information. If you have a block there, things happen, and if left unrepaired, the DNA itself can break. And if a change in a single letter can cause cancer, let alone loss of chunks of information. So if the repair is important, because interstrand crosslink, actually, I won't go into that, but it can be caused by multiple causes, whether external or internal. The cells need to be able to repair this. But how do they do it? In general, it goes like this, in this particular manner. So that ICL over there, you have ICL, and the cells need to, using sensor proteins, sense or detect the ICL. And then what happens next is this ICL gets cut and then removed, and then the gap over here will be replaced by new strands of DNA, newly synthesized strands of DNA carrying the correct code. So in general, that's how the cell do it. But again, how do the cells, like the proteins, the sensor proteins, can recognize ICL? Because these proteins, remember, they don't have eyes, and yet they have to detect specifically spots of ICL in long strands of DNA. How do they do that? In general, there are two categories of strategy. One, replication-independent repair. Secondly, replication-dependent repair. So, with the first category, which means the cells don't have to do DNA replication to detect ICL, what happens is, if you have a certain region, again, which is accessed or transcribed quite frequently, when you have a block in the transcription, the cell recognizes this and it attracts and recruits sensor proteins and effector proteins, the red ones that function to repair the lesion. Or what if the region where the ICL forms are sparsely transcribed or sparsely accessed? Remember that I showed you the diagram before. Maybe I didn't. Sorry, I didn't emphasize on that. But ICL, interstrand crosslink, when it forms, it distorts the DNA. So it actually changes the shape of the DNA. And certain proteins can recognize that and then repair it. The second strategy is when you have replication-dependent repair, which is when the DNA replicates its DNA, like synthesize new strands for its children, the presence of ICL block that. And then the cell again can detect this, and it results in repair. Okay, now, but the question is now, both of these repair mechanisms are important. The one, number one, the former one, especially when you have cells which are not dividing, like neurons. They can't divide, so you have to detect it in the absence of replication. But sometimes you also have cells that divide rapidly, so they have to make use of this process. But the question remains, whether these two modes of repair, are they overlapping with each other? Whether they have the same sensor proteins or not. So this is my strategy to identify, to elucidate the mechanisms better. So first I treated my cells that I grow in the lab with ICL-inducing agent, basically. The function of this is to enrich the repair proteins and activate them inside near the DNA with crosslinks. And then I extracted, using various biochemical methods, extracted the proteins. And then I do what I call fishing. So I have these probes, like DNA, I synthesize this DNA with different conformations. So that one is linear or straight line, that one is it mimics replication fork to be, mimics replication fork that is formed during DNA replication. So then I mix the protein with that probe, and then my aim is to capture certain proteins that, for example, if the protein is involved in replication-independent ICL repair, it will bind this one. But if it is involved in replication-dependent ICL repair, it will bind to this one. So in this way, I can separate the two processes. And then what I do after that, I capture basically the probes with the proteins attached, and then I can analyze what is present in the sample. For example, here, if the linear DNA, the probe does not contain protein, it will not contain anything, like very minimal amount. But when it has ICL, for example, I will have one protein that is dominant, let's say, or enriched, we say, and then I can identify what that is, and then work with that. And then, with that, I'd like to thank my lab, and that's all.
Okay, keep the Q&A for the next speaker, because we already have a break after the first speaker is open. So the next speaker will speak and then have a break. So yeah, the next speaker will continue there.
I'm sorry.
Hi everyone.